Hello everyone. So today in this video we are going to study about cell organelle and cell inclusions. We will also include one of the organelle, and that is endoplasmic reticulum. Now, to understand what are cell organelle and cell inclusion, we will have a brief study about its characteristics. The first one is that these organelles are subcellular structures, right, which have their characteristic forms, structures, as well as functions, right. And these organelles, they are specifically found prominent in eukaryotic cells, right. And the next thing is that they have membranes present around each and every such cell organelle. It can be a single membrane structure, it can be a double membrane structure accordingly. Next is few examples, right? We know these examples already. We have studied about them. We know about these structures like mitochondria, endoplasmic reticulum, Golgi apparatus, chloroplast, etc. and etc. Next is inclusion. What are inclusions? These are some non-living structures, right? And they are present in different forms within the structure and help in storing food. It can help in storing secretory material, excretory material, pigments, etc. These type of materials can be stored actually within vesicles. So vesicle within a cell act as inclusion, right? So these are non-living and they help in various way like storing food, secretory material, pigments, etc. So examples like vacuoles, cell wall, protein, if they are contained within cell inclusion within this vesicle like structures, resins, latex, if they are stored within vesicle like structure, then these are known as inclusions. Next, we will study about endoplasmic reticulum. Here we can see the figure, the diagram of endoplasmic reticulum attached with nucleus. You can see the structure of nuclear pore, the nuclear envelope, and the entire endoplasmic reticulum. This endoplasmic reticulum is highly complex network of membrane bound tubules, vesicle, and sac-like structures also. So you can see the structure given in the figure there. Various inner folded membrane like structures, tubule, it can be vesicle, it can be in the form of sac. So these are the way how the structure gets formed. And there are also two types of endoplasmic reticulum according or on the basis of its formation. You can see very clearly that the closer one on the diagram, the closer one toward your side, which is closer to you, the one which is having ribosome, those dotted like structure, ribosomes attached with it, these structures are called as rough endoplasmic reticulum. So ribosomes, we know that these structures are protein factories of the cell. They help in formation of protein. And if they are studded, if they are attached on the wall of endoplasmic reticulum, that becomes rough endoplasmic reticulum. So they can also help in protein synthesis. Next is smooth endoplasmic reticulum. On the other side of the figure, you can see their shapes. they are not studded or attached with ribosomes and hence gives a smooth appearance. And that is why the term smooth endoplasmic reticulum. Just because they don't have ribosomes attached on their wall, they don't help in formation of protein. Rather, they help in formation of lipids. Now, let us see the functions of endoplasmic reticulum, right? The first one, rough endoplasmic reticulum, helps in protein synthesis, as we already came to know that rough endoplasmic reticulum is known as rough because of the presence of ribosomes, and these ribosomes help in the formation of protein. The next, SER, smooth endoplasmic reticulum, helps in lipid formation or lipid synthesis. Lipid means other name of fat. Now, as these protein synthesis taking place, as this lipid synthesis taking place, it gradually gets filled within vesicle-like structures and moves out from endoplasmic reticulum towards the membrane and helps in the formation of membrane. So that is why this process is known as membrane biogenesis. Bio means living. It is forming out of living parts of the cell, and genesis means production. So the formation of membrane with the help of living component, that is why it is named as membrane biogenesis. Next is it helps in intracellular as well as intracellular movement of substances or transportation of substances. That means any molecule, any substance which is being produced inside the nucleus can move into the cytoplasm, and that can be through endoplasmic reticulum. It can also have the movement of molecule from the cytoplasm to outside through the help of endoplasmic reticulum. So that's why intracellular as well as intercellular movement or transportation of substances. Next, if we see the entire structure, you have seen the structure is highly folded into tubule, into cisternae, right? So just because the structure is highly folded, that's why it provides a large surface area, and hence various chemical processes can be produced, can be undertaken. So that is how production of molecules, production of substances are helped through this large surface area. The last part of it is that smooth endoplasmic reticulum, the one which was not having ribosomes attached on their wall, that helps to take part in detoxification, helps to detoxify all the toxins, all the harmful chemicals that might have come across the membrane and got entered into the cell. So this particular part of the cell, smooth endoplasmic reticulum, takes part in detoxifying them, detoxifying harmful chemicals, drugs. Clear? So this is what, with this we come to the end of the discussion on endoplasmic reticulum. Thank you everyone.